Humans have a unique capacity to override primal instincts and make difficult long-term choices. This ability originates in the prefrontal cortex, often referred to as the brain's personality center. It is the cortical region responsible for higher-order executive functions. The prefrontal cortex is unique because it is neither a purely sensory nor a purely motor component of the brain. Instead, its power comes from acting as a massive integration hub. Looking at this map of the prefrontal cortex, we can see exactly how the frontal lobe is structurally divided. The topmost section is the dorsolateral region, shaded green here, which dictates planning, strategy, and executive decisions. Below that, the ventrolateral and orbitofrontal regions, marked in red and dark red, serve a different purpose. They actively inhibit primal survival responses arising from the limbic system. The limbic system is a complex network of deep brain structures that controls basic emotions and drive. These distinct areas constantly synthesize in-the-moment information by maintaining reciprocal connections with the rest of the brain. Without this specific biological structure continuously integrating data, humans lose the foresight and social adaptation required to navigate complex interactions. Our understanding of this region advanced significantly following a serendipitous medical case in 1848 involving a man named Phineas Gage. During a railroad construction accident, an iron tamping rod was driven completely through Gage's skull and frontal lobe. The physical trauma was heavily localized to the orbitofrontal lobe. While his basic motor and memory functions remained surprisingly intact post-accident, his personality was entirely altered. A once responsible man became prone to volatile behavior and completely lost his capacity for social regulation. This specific clinical outcome initially revealed the division of labor within the prefrontal cortex, demonstrating that planning and impulse control are handled by entirely different local networks. Gage's survival proved that personality is mechanically rooted in the physical integrity of the prefrontal cortex. The structural origins of the prefrontal cortex begin early in the development of the central nervous system, around the 4th week of gestation. Development starts with a flat neural plate that folds inward to form a neural groove, which eventually pinches off to create the closed neural tube. This diagram outlines how the front of that neural tube, the prosencephalon, bulges laterally and differentiates into the telencephalon. The telencephalon is the embryonic structure that eventually matures into the entire cerebral cortex. During early childhood, the prefrontal cortex experiences an initial massive overgrowth of neurons and synaptic connections. Pacing quickens slightly. This rapid expansion is immediately followed by an extensive, highly destructive phase known as synaptic pruning. Synaptic pruning is a natural biological process where the brain eliminates weak or redundant neural connections to increase network efficiency. This aggressive reduction of connections is precisely what carves out delineated, functional pathways for executive control. Developing executive function requires the aggressive, precise elimination of excess neural noise. Maintaining this dense, energy-hungry network requires a massive and highly specialized vascular supply. Returning to our anatomical map, we can see the lateral and anterior aspects of the prefrontal cortex are supplied by the middle cerebral artery. Conversely, the superior, medial, and orbitofrontal surfaces draw their blood supply from the anterior cerebral artery. For decades, it was a standard medical assumption that the central nervous system lacked true lymphatic vessels for waste clearance. Recent evidence has updated this model, revealing the presence of a macroscopic waste clearance pathway called the glymphatic system. This glial-dependent system utilizes perivascular spaces, the fluid-filled areas surrounding blood vessels, to facilitate lymphatic drainage. Waste flows from the meningeal sinuses and deep cerebral veins, eventually draining directly into the cervical lymph nodes in the neck. The highest order cognitive functions rely completely on this highly specific, dual arterial plumbing and complex waste clearance system to stay operational. Unlike many other cortical regions, the prefrontal cortex receives zero direct stimulation from peripheral nerves. This diagram illustrates how the prefrontal cortex relies entirely on large association bundles, known as fasciculi, to connect with primary sensory and motor cortices deep within the brain. These specific nerve tracts act as high-speed cables, supplying the exact in-the-moment data the prefrontal cortex needs to process our surroundings. When this structural volume is malformed during development, the consequences are severe. In holoprosencephaly, a developmental disorder where the brain fails to divide into distinct hemispheres, prefrontal cortex volume is drastically reduced. Beyond structural malformation, physiological variants in functional activity correlate tightly with clinical diagnoses. Decreased activity within the prefrontal cortex has been consistently revealed in depressed patients. Similarly, reduced interneuron cortical inhibition, which leads to hyperactivity in these networks, is strongly associated with schizophrenia. Major psychiatric and neurodevelopmental conditions are directly observable physiological variants of the prefrontal cortex's wiring and regional activity. Because the physical pathways are tightly correlated to function, frontal lobe neurosurgery carries exceptionally high stakes. Surgeons utilize motor and language mapping in awake anesthetized patients to navigate this terrain safely. These intraoperative maps allow surgeons to meticulously avoid causing the dramatic neurologic deficits associated with damaging the prefrontal cortex proper. Looking closely at this anatomical map, we trace clinical outcomes of damage to the lateral prefrontal cortex. A lesion in the dorsolateral area causes catastrophic loss of working memory, the system for temporarily holding immediate information. These patients cannot perform delayed response tasks. They also show severe impairments in recalling the specific context in which a memory was originally founded. Structural damage to the dorsolateral region breaks an individual's mechanical ability to hold, sequentialize, and contextualize immediate information. Shifting to the lower sections of this model, lesions in the ventromedial and orbitofrontal regions present different clinical outcomes. Damage here causes severe difficulty with cognitive task switching, creating a cascade of behavioral failures, including poor judgment, rapid, exaggerated changes in mood, and inappropriate affect. These structural lesions are frequently compounded by the dysfunction of key neurotransmitters housed within these pathways, specifically dopamine, norepinephrine, and serotonin. These specific structural and chemical dysfunctions map directly to the broader clinical landscape of neuropsychiatric disorders, including bipolar disorder, severe obsessive-compulsive disorder, and attention deficit hyperactivity disorder. The prefrontal cortex is a meticulously mapped, biologically vulnerable machine,